I am Dr. Didier Montet, and I am working in Montpellier, in the south of France, in an international research center, which is CIRAD. CIRAD is an international center that deals with the developing country. The aim of my team is to help developing countries from Latin America, Southeast Asia, and Africa to develop the quality of their food. We are working on mycotoxins. It means fungi that produce mycotoxins, which are very toxic and produce some cancer in people. And the main products that we are working on are coffee and cocoa. When you try to analyze the food in tropical area like this, because of the humidity, because of the temperature, there are always a lot of microorganisms. For example, on a fish, on an aquaculture fish from Vietnam, you can find more than 100 strains of bacteria. If you want to analyze this bacteria, it could take a long time in the laboratory. It's why we developed 15 years ago techniques that permit to analyze in one step and in less than 24 hours all the bacteria. And now we could analyze bacteria on fish, on fruits, on seeds, on what we want, on all the foods that are possible to export from this country to European Union. At the beginning, we were working on bacteria, then we developed this technology for yeast, and then we developed the technology for fungi. The name is PCR-DGGE, which means PCR is the multiplication of DNA of bacteria or yeast and fungi, and the DGG is a special gel that permits to separate the DNA of the different bacteria. The PCR that we develop is a PCR that uses a non-specific primer, or it means that this primer could amplify the DNA of all the bacteria in one step, with recognizing a specific part of the bacteria. It means that all this part of DNA have the same size in terms of pair of base, and it means also that we could not use the gel that were used at this moment 15 years ago, you could imagine. And we had to develop a specific gel that could chromatography and separate the DNA with the same size. Well, the advantage of the system, when you see the gel, you can see the food, you can see the bacteria, and also we can extract the part of the DNA that is on the gel and sequence this part of the DNA and then get the name of the bacterium. And it is the same also for fungi. When you analyze the seeds, you have the toxinogen fungi and the non-toxinogen fungi. We are using this non-toxinogen fungi now as competitor of the toxinogen fungi. That's a new development and we are working on. The biological bytecode is the photo of this gel. You could imagine bands of DNA and you take it as a code, as you can find on a cheese, on a bottle, things like this. And you take a photo of this code, and then you could compare this code with other code. And when you compare this, the code of a fruit with the code of the same fruits cultivated in another country, if you compare them, you can link that to the origin of the fruits. That's the advantage of the technique. It means that we created at this time the first tool that permit to link the ecology of the fruits or the fish to its geographical origin. We are discussing about regulation on food safety in European Union. And this food safety regulation was published in 2005. Because the regulation was published without any tools, scientific tools, it means that actually if you want to follow a food on the earth, you have to follow the stamp of the food. Only the stamp and the administrative paper. We have no scientific tools that permit to say, okay, this food is coming from US, this food is coming from Ireland. That is not possible actually. And the objective now of the scientists is to develop analytical tools that could permit to follow the regulation. With this type of research, we could improve food safety. Because if we know, for example, that in some country they produce nuts that contain ochratoxin or aflatoxin that are very dangerous for human beings, with the analysis of the origin, we can know if this product could be dangerous or not, and then analyze specifically the hazard.